always a full-fledged dementia. In fact, we'll argue in a few moments that the dementia is actually the least common of the three. This FTD, MND-like component, these are actually cases where an individual has passed away with ALS, an autopsy has been done, or they've passed away with a dementia and an autopsy has been done, and we see that they have features of one or the other that we didn't see during life. And so that's a neuropathological one. And then this final one at the bottom here is ALS dementia. And this is something that we're increasingly seeing and being aware of, is that having one neurodegenerative process does not preclude you from having another one. So you can have Alzheimer's disease, and we'll see it's anywhere from 1.5% to 2%, or Parkinson's, depending on where you are in the world, and there's a dementia that can be associated with that. So this ALS dementia is meant to capture everything else. So these criteria were published in 2009, and were applied over a number of studies. And here I'm showing you a study from Julie Foucan and her group in 2012, where these criteria were actually applied to a large cohort of ALS patients. This is 160 patients with ALS. And the first thing that's most important here is that this 47% is that percentage of patients who have no other abnormality, right? So a little around a half of the patients are normal in this particular study. As we start to look more carefully, 14% of the patients actually had the ALS FTD. Remember, what I told you was that Arthur, almost 20 years earlier, in fact almost 30 years earlier, had predicted that 15% of ALS patients would have a dementia for it, and here's the numbers now in real time for that. This 2% up here is, as I mentioned earlier, those individuals who have an Alzheimer's-like, but then the rest of this population here, in this particular study, making up somewhere around a third of the remaining patients, have the more subtle invariants, and we'll come back to what they look like again. Now, this isn't the only study to have done this, right? So now there's been several of them. So if we look across the literature, the reason why I've selected these three studies is that these look at everything from Asia through to Europe through to North America. And the data is the same over and over again. So somewhere around a half, it's variable, can be as high as a half to 60% are normal, but then half of patients, when we start to study them carefully, have a deficit. And the true dementia may be from 5% to as high as 28%, depending on how they're studied and the criteria being used at the end of the day. So this is not a curiosity. This is not something that is rare. This is something that is actually, when one looks carefully at ALS patients, somewhere around a half to 60% will be affected by this disorder. Now, clinically, this is important, but can we actually look at this and see any imaging changes? And indeed, we could spend the next hour looking at imaging correlates to this, and there are a number of studies which are superb in the literature. I've selected one from a colleague in Italy who looked at something of a way of us to look at the surface of the brain. So this is called free surfer technology. And in essence, what happens here is, imagine that you had in your hand a walnut, right? And just picture you've taken the husk away and you have that pure walnut sitting in your hand, and it has all these grooves and marks and everything over it. So it kind of looks like the surface of a brain. Now, if I were to say to you, take that and smooth it out so that it's no longer got all those ruffles and ridges, but rather is a nice flat picture. That's what you're looking at in free surfer. Okay, so the brain doesn't look this smooth, right, as you're seeing it here. They've flattened it out. But what they've then done is they've looked at areas of cortical thickness. And cortical thickness for us is a measure of the health of those regions of the brain. So a thin cortex suggests that there's been some neuronal loss to it. We also look at how much fluid there is surrounding the brain. The smaller the brain, the more the fluid. The areas where there's atrophy, you're more likely to see more fluid going on. So there are very ratios that you can look at here. And you'll see over here on the right-hand side is basically their gradation. So the more intense the color, the greater the loss of cortex, so the thinner the area for it. So for those of us who work in the field of ALS and such, we're not surprised to see this. This is an area of motor strip. We're not surprised to see this as well. That's where a lot of motor innervation comes from that affects the limbs and the trunk of the body. we expect in a motor disorder where the neurons at the surface of the brain are being lost that there will be thinning of that region of the cortex. No surprise. But look at this. Massive areas, and this is on the inside aspects, what we call the mesial aspects. So this is mesial forebrain or frontal lobe, and we're seeing significant patterns of atrophy already there. Equally as important, as we look here in the more frontal and some of the temporal regions of the brain, we're also seeing significant atrophy. These regions have nothing to do with motor control, right? Or at least the motor innervation pathways themselves for it. They have a lot to do with some of the planning aspects, and again, we're going to return to that in a moment. So here we have evidence using the criteria as they were published in 2009, and then also on imaging, and as I mentioned, multiple studies which clearly demonstrate we have pathology and changes in the brains of individuals with ALS well outside of areas where we would have expected to see it occur for that. Now we met 2 years ago as part of this every 2 years sort of group and looked at these criteria. And it was clear that a couple of things were happening. And one is that whilst the criteria were very robust, they were not easy to use. And in fact, we needed to simplify these criteria and make it easier for clinicians to use in their outpatient settings. I can tell you we're doing a study, as David had mentioned, right now in Ontario, that we're looking really, really critically across a number of disorders, including ALS, for cognitive deficits. And this is up to a 4 to 4.5 hour study, depending on the types of pieces that we're doing. Well, you simply can't do that in a clinic. So we need things that are going to be easier. So we looked hard at this and revised the criteria, the international one. And we recognized that we need to have criteria that are easily used in clinical practice, that don't involve a lot of research tools. But we also need to understand these disorders far more clearly. So there are certain groups of studies that are much more research intensive. And so in the end, what we suggested was that we would have 3 different levels of testing that would be available and to be utilized. And the easiest one is what we call level 1. These really don't require a lot of training. They've been well-validated studies. They can be used for clinical research. There's no biomarkers that we're going to use with those. And we're using it largely for diagnostic purposes when imaging is done, as opposed to looking at other features in there. All the way up to this level 3, which is very complex studies, really should be done as a research protocol. Use biomarkers to try and understand if there are ways in the blood or the spinal fluid that we can make analysis, and then very intensive imaging studies. And as you might imagine, at one end, this is fairly inexpensive. As you get up here, this is very expensive to do. So these are research tools in here. So one of the major advances in the course of these last criteria, and they were really just published 6 months ago, has been to say we recognize that the previous criteria were difficult to use and required a whole team to be able to administer. These new ones are easier. So why did we also need to change things? Well, one of the reasons is that we began to see that over time, and using these criteria from 2009, we were starting to see clear patterns of deficits when measured at the bedside that were consistent across these patient populations. And so here in individuals who have cognitive impairment, things such as verbal fluency turned out to be one of the most solid indicators that there was a global cognitive impairment. And what do I mean by verbal fluency? Well, I hope that what you're hearing, if your connection's working well, is that my speech is relatively fluent, that there are not a lot of hesitations in it, that if I make word-finding errors, that they are kind of expected ones that you would see from time to time, that my sentence structure is relatively intact. Not always perfect, but relatively intact for it. So that's verbal fluency, and that requires regions of the brain where I showed you on the imaging before, where we didn't expect changes to occur, to be functioning very well for that. So this is a study out of a group, again, in Europe, Refort's group, but several years later, he and his team returned and looked at the same measures again. So this is a study that just came out last year, but we saw the early data presentation when we were redefining the criteria, and in essence, this up and down line here is if the abnormality had no impact whatsoever on your diagnosis for that. Everything to the right-hand side means that it had a positive influence. So if we look again, here's verbal fluency. You can see that across a host of studies with all their data brought together, verbal fluency turns out to be a major abnormality in patients with ALS. But so is executive function. And things that we were surprised to see, even memory, we're starting to see come forward as that there are abnormalities in here for that. So depending on the region in which you're studying and looking for clinical characterization, really nice data that is suggesting that there are impairments that we need to pay attention to. This concept of behavioral syndromes in ALS, also very, very important. And I don't know as a clinician how many times I've sat with families and loved ones and had a conversation around the change in behavior of a patient that we're looking after who has ALS. And it can be everything from an enhanced sense of irritability to what appears to be apathy, to inappropriate laughter or inappropriate behavior, decision-making processes. And the conversation 25 years ago when I started doing this was, well, we think it's just a reflection of having the disease. Well, in fact, it may not be that. Here we see in a study of patients across several different studies that apathy is a major feature of patients with ALS beyond what you would expect to see simply because having the disease alone. So things like loss of sympathy or empathy, changes in eating behavior, lack of response to social clues, poor decision making. Right? I know a lot of families and patients are on the line right now for this. Well, the number of times that I present this to my own patients and their families, and they go, that explains something that we just thought was a little bit odd. Maybe there's something going on here. As a clinician, that then suggests to me, okay, I need to do a bit of testing here for that. And there are certainly tests that can be done relatively easily at the bedside to sort out whether this is a process or not for it. We have to be careful, right? And this is one of the things when I'm lecturing about this elsewhere in the world, is to be very, very careful that we don't throw everything under the bus, so to speak. If somebody has apathy, we will also find impairments in other areas. So everything from executive function, right? So if they have apathy, it will be worse than what you would expect to see if they have dementia at play. So the apathy, in fact, itself will contribute in some ways to the scoring, or the alternative is that it is an additive feature of the disease itself. We just need to be careful when we're interpreting that for it. Perhaps the biggest change, however, is something that we didn't even know about when the first criteria were coming through. Now, to say we didn't know about it is probably an oversimplification. There have been a lot of work talking about something we call executive function that has, how does it link to what we call theory of mind. So what does theory of mind mean? So this is a picture I use and frequently when I'm giving this talk. And these are two individuals from Safari when I was traveling on this that I took a photograph of, and they are a long ways away from me. But what do you see in their faces? And I think everybody at this point would say that there's sheer pleasure, sheer joy in here. Well, I don't know that. Right? They're a quarter of a mile away from me, and I'm photographing them through a telephoto lens with all of this. But what you've just done is if you've used your own value system to imply, based on that facial expression, that they are happy for that. That's the first level of theory of mind, your ability to recognize in others an emotional response and to understand that. So can you imagine if you were an ALS patient and you had impairments in here, and then you were a caregiver, and you're trying to understand how is it that my loved one can't understand what it is that I'm trying to present or the difficulty that's occurring here for it. So that's the first level of that. The second level would be for me to ask you, so why do you think they're so happy for that? And this would require now that you take your value system and your understanding, you apply it to there and say, well, maybe they're just coming to join their village. Maybe they've just come from a wonderful experience. Maybe they're sisters and they've seen that there's a group of us who have come to join them and have a conversation. There could be hundreds of reasons for all of it. But now you're moving beyond the simple recognition of what's going on to actually stating, now I think I understand why this is happening in there. And again, if you were an ALS patient and could not do that and understand why your loved one might be upset for it, it's going to create a very difficult dynamic. So this is an important part of just day-to-day understanding. How often does it occur? Quite frequently, and we'll see that in a moment, but these types of abnormalities are telling us about theory of mind arising from different regions of the brain. And it's not important that I tell you exactly what the prefrontal cortex is in this, other than to say that this is the region that I was showing you so severely damaged with cortical atrophy in that earlier image from Adanzato's group for it. So we're now starting to understand when we have that cortical thinning going on, it's actually being reflected in this inability to understand other individuals' mental states based on your perception of those. Very important piece in there. Now, if we actually start to look, and this is a study that Sarami and our group has done, and looked at individuals with ALS who have no evidence whatsoever of frontal temporal dysfunction and have neither of the two most common mutations that we would think would be related to that type of dysfunction for that, and almost half of them already have difficulties in what we call emotional attribution, so a link to that theory of mind for that, and they have loss of the gray matter, so thinning of that cortex. So this is before any other problems are at play, and we're seeing this in hand. A slightly earlier study looking very similar again, 18 ALS patients with no evidence of dementia, 50% of them have abnormalities already consistent with dysfunction in the regions of the brain that I was showing you. And remember, I said somewhere between 50 and 60% of patients are predicted to have a deficit that will be frontotemporal in its nature. So very important that these studies are saying to us, don't make the assumption that an individual is normal and has no deficits if you haven't tested them. You need to actually spend some time sorting this through. Here's the regions again, right? Social cognition. So if we go back to Adanzato's, this is the area of the brain that we're talking about, right? And this is the area, so motor strip is up in here for us. This is the prefrontal cortex, more mesial aspects, and this is where we're seeing deficits in these patients. So it's beginning to line up very nicely. And here, if we look at an individual who passed away with ALS, who had one of these syndromes, you can actually see the region of the brain here where that atrophy has occurred, whereas more posteriorly, actually where motor is, not so much change. This is really in that frontal regions for us. Now, what about this issue of memory, right? It's a little early days for us to know, and so in the new international criteria, we've been a little cautious on this, and we'll probably evolve in about 5 or 6 years to including it more. But nonetheless, as we start to look carefully, there are individuals who have difficulty on immediate recall, and it can be as high as 1 in 5. When we look with more functional imaging, areas of the hippocampus, as we see here, which are important in memory registration and retrieval, there's evidence of loss of muscle or of loss of bulk already there. So we're beginning to see this early anatomic and clinical correlation on memory. So I don't think the story is completely done yet, but I think we're pretty close. So where are we now, right? So this is the revised international criteria. These just came out. And again, this is really important for you to understand that in the absence of the ALS Society of Canada coming to the table, and for the Windsor chapter and the Michael Halls, we wouldn't do this. This conference brings people from around the world to have this discussion over the course of 3 days. And these are the new criteria. And there's a couple of real significant advances here for us. First one is we still have sporadic ALS. This is very much what Charcot described, a pure motor disorder. Familial ALS. We are now suggesting that this should be called genetic ALS. And other speakers in the course of the next day and a half will be telling you reasons as to why this is so important. But the bottom line here is that as we look more carefully, we're beginning to see individuals who have ALS with no clear evidence of a family history for a host of different reasons, where we're finding genetic abnormalities in them. It's not a major component, but nonetheless, it's more than 0. And so we need to be thinking about, are the genetic causes of ALS far more widespread than we think, and how do we actually deal with that? ALS FTD is still there. There's the behavioral and cognitive component, and now we have where you combine both ALSCI and ALSBI together, and this gets us the new theory of mind and dysexecutive. And the breadth of changes in the FTD and behavioral ones are much larger now. So the criteria have expanded. We think that they're going to be much more useful for patients, but specifically for researchers and clinicians. Now, why is this so important? Well, as my opening slide said, this is more than just a curious finding. And this, if I spend a minute or two on this in the next slide, is the reason why it still puzzles me that when worldwide we clearly recognize that these syndromes exist, and yet we do not pay close enough attention to them either in the design of clinical trials or, as I say, even in some of the most recent academic processes, whether it be a meeting or publications, and these are completely ignored. Here we see executive dysfunction. Okay, so executive dysfunction is part of that cognitive impairment paradigm. So talking again about that frontal regions of the brain being involved for that, and individuals who either have executive dysfunction, right, or have other types of cognitive impairment that do not have the executive components to that. And immediately what you see here is that this is a survival curve. So this talks about from the time of symptom onset, measured in months. Over on this side is at time 0, you would have 100% of people alive. And eventually, unfortunately, because of the nature of this disease that we work in, people will die, and they will do so at a predictable rate in it. So these are individuals who have no evidence of executive dysfunction. So on average, survivorship somewhere between 3 and 5 years, exactly what we teach with that. But look what happens if you have executive dysfunction, so evidence of that frontal region involvement of the brain. Almost 1 year of shortened survival, if this is present or not. And this has now been replicated in a number of studies, and we do not understand fully why yet this is the case. But this is a very critical point in here. If I told you that this was ALS, and this was ALS treated with a drug, we'd be through the moon, right? This is a significant difference in here. And if I had a drug that made a difference in this survival of this magnitude in here, we would be ecstatic. That would be a home run for us. It's not a cure, but it would get us started on that. And yet here we have a variable that tells you right away that if you have this executive dysfunction, it's actually going to be worse in terms of survival. Now, why is that so important for us? Well, the very first thing is, can you imagine if I set a drug study together and I had 1,000 individuals on medication, and 500 of those individuals were receiving placebo, and 500 of those individuals were receiving the drug. And somewhere within that group that was receiving drug, the majority of those individuals had executive dysfunction, and we didn't measure it. One of two things would happen. If the drug had any benefit at all, and let's say it gave a 1-year survival benefit for it, you would not see that because it would be lost in the analysis, because those individuals who were at a greater risk to die within 1 year earlier were not taken account for. Right. Alternatively, you could actually have a drug look worse because it had actually showed a large population who was doing worse, and yet you wouldn't have explained it on the basis of a very specific phenotype sitting in there. So the ability to look and to include evidence of frontotemporal dysfunction in clinical trials is critical. And it's one of the reasons why when we rewrote the criteria, we wanted to make sure that we included testing paradigms that could be easily added to any clinical trial. And quite frankly, from my vantage right now, having done this for, 27 years now, I think actually there is no such thing as a clinical trial now that should be done in the absence of this as a measure. It just is beyond comprehension to me. Here it is, and individuals who also have abnormal behavior, even more striking, right? Individuals who have a dementia paradigm, and this comes from a Western Pacific study, so they tend to still use the term dementia with this, but if they have abnormal behavior, look at the difference in survival already. So we cannot ignore this. It is a really critical aspect of the disease process itself. What I want to do is just spend 1 or 2 minutes and just tell you a little bit about potentially how this might be treatable. Now, I'm going to have to restart the program. David, are you still there?
I am.
My PowerPoint just says I need to restart my program. That would take a minute or two.
Okay.
But the other question, that's okay? You're good?
Sure. Yeah, we're good for time. No problem.
Okay.
So if you want to unshare your screen, yeah, go ahead.
Yep, and it's just going to bring up the program again.
Sure.
And we'll scoot right along. Isn't technology a wonderful beast?
Well, last year my computer crashed around the same time of the day, so.
Oh, really? Okay, are you seeing a screen now?
We are.
Oh, perfect. Okay, so in the last couple of minutes, what I want to do is actually give you something that's a little bit on the hopeful side of all of this. And this, you know, when we go back to the Ice Bucket Challenge and the type of assistance that's been brought forward to help us understand and move these, you know, characterize this basic cellular biology of ALS, this is an example of where that's been an absolutely critical piece for us. The studies that were done here were done really because we had seed funding that came from that. So the bottom line here is that we spent a great deal of time over the last 2 decades trying to understand what's actually happening to those cells within the region of the brain where we saw that atrophy. So going back to that early slide on the free surfer where we said there's thinning of the brain. And we looked under the microscope, and what we were seeing is within neurons, and this is what are neurons within the brain, and these are cells that are supporting cells called astrocytes. They're stained for a protein called tau. And this tau protein is depositing within these cells, and it simply should not be there at all. It should be sitting out into processes because it's an important structural protein. But this type of an aggregate is not normal. Typically, it would be seen in Alzheimer's disease. But we proved by isolating that protein and actually characterizing it carefully that in Alzheimer's disease, it has a very specific type of characteristic. This is not our work, this is, I mean, from our paper, but this has been known for decades. But here we see exactly the same type of isolation in ALS with evidence of cognitive impairment, and it looks completely different. This is not the same as this. So right away, sometimes we get a little criticism for this, and people say, well, it's just Alzheimer's disease. This is not Alzheimer's disease. This is a different entity entirely. And in fact, when we began to look more carefully at the nature of the protein itself and how this tau was modified, we came across this region of one very specific, what we call an epitope, which is changed in its phosphorylation state. So imagine, if you will, I gave you 10 pages out of a telephone book. And I told you that in that telephone, the 10 pages that you've got there, the entirety of all of those telephone numbers is important for how that telephone book works. And you can mess around with a couple of those numbers, and it's not going to make a great deal of difference. But there are a couple of the numbers you don't want to mess around with at all, because suddenly things don't read right, or the pages stick together, or suddenly you get big blank areas for it. And so proteins are like that. They're large telephone books of information, and you can take little changes, and sometimes they're fine. Sometimes you make a change like this threonine 175, and you put a charged group like a phosphate onto it, and it's not a good thing. We actually created a mechanism whereby we could actually see these proteins. So the 175, something that we thought was abnormal about tau, we created what's called an antibody, and that's simply a way for us to look on tissue sections and to see whether there's an abnormality there or not. And here we see that in patients with ALS with cognitive impairment, we get these really fascinating inclusions in what we call dystrophic neurites that should not be there. This is Alzheimer's disease. Some of the cells we see it as well in Alzheimer's, so it's not absolutely unique to what we see in ALS, but in ALS with cognitive impairment, 100% of the cases will have this. Even in patients who do not yet have cognitive impairment, we were finding early neuropathological changes. So this simply tells us that it's there. This was very controversial, and I think it would be fair to say remains controversial in the literature, because this would define ALS as something we call a tauopathy. And quite simply, that's not something that's viewed to be traditionally correct. It's always thought that if there was a dementia, for those who still don't believe it, in ALS, that it wasn't a tau process, or that if there was a tau deposition, that it was just a side effect to it all. But this is an important study here from last year, and this is David Mann's group out of England, who said, well, listen, somebody's got to look at this again, because if Mike Strong and his group are correct, then it is a fundamental shift in our thinking, but somebody'd better look. And the bottom line here is that they found the same percentage of ALS patients with tau deposition as we did. Slightly different interpretation to it, but at the end of the day, definite tau deposition. Now, why is that so important? It's critical because we can actually go into cell culture, and what you're seeing here on the right-hand side are cells growing within a dish in which we have placed into those cells a genetically modified tau protein, which is human, which has that abnormality that we see in ALS. In those cells that have no abnormality of the tau, it's just human tau, you see this beautiful fibrillar network. This is what it should look like. Within 24 hours, if I put the ALS with cognitive impairment tau in, look at these fibrils that are sitting here that are not normal. And this is the beginning of those aggregates that we see in the human brain of individuals who have died from this. And indeed, if we look at markers of cell death, by 72 hours, these are the cells here in the gray that express the human tau that is abnormal. So we know that not only do they form these fibrils, but they begin to kill the cells with that. It was important, therefore, to begin to understand, is this something that could actually happen in a whole brain? Was this just an artifact, or was this an important feature of it? And so in experiments that we're just starting to publish right now, so this is data that you're getting to see for one of the first times, is that we then said, well, we need to create a way of putting the ALS tau from those with cognitively impaired into a rat brain. Now, the rat brain is important because it has a lot of structure that looks very, very much like what we see in the human brain for that, including the hippocampus, which is this structure here. And so we used a virus, and we genetically modified that virus, and we injected it neurosurgically into this region of the brain. Here you can see a little hole left over from where we injected it. And the virus has a way for us to track whether it's in the brain or not, and that's this brown staining here that you can see. However, after 3 months, even as early as 1 month, in cells that are sitting within that region of the brain where this is being expressed, we are beginning to see the formation of these aggregates. These are tau fibrils that do not belong. These are the same things that we see in the brains of individuals with ALS who have cognitive impairment for it. So this, in essence, proves that this abnormality is not a side effect. It, in fact, when present, is sufficient to drive the formation of this pathology. And we know that this pathology, from the experiments I just showed you, will cause damage and kill the brain. Now, that would be one thing, but being a clinician and a clinician scientist in this, the idea is, how do we treat this? Right? At the end of the day, we need to be able to put a stop to this. I'm not going to go through all the details of this. It's important to know simply that we now know how to go from here, which is the normal, to this pathological state. And we know every step that is required to do it and what is triggered by this change at 175. But what we now know is that if we use a series of drugs, and here we see the survival of cells, so this is the number of cells that die or have fibrils within them, so this is cell death, and this is fibrils. And here's the ALS change. These two are controls, so expect what you expect here. But if we block this pathway, we completely inhibit not only the formation of fibrils, so you get none of this happening, but you also completely block the cell death. And one of the drugs we've used is lithium. Right? Now you might say, well, wait a minute, didn't they use lithium in treating ALS? Well, the answer is yes, it was, but it only studied the motor function. Nobody looked at the cognitive in detail sufficient to know this. So we know that we can pharmacologically now block this. And we know that this is important because it's all happening in the same cell. So it's not that there's some sort of an artifact going on. So at the end of the day, why is this so important for us? Okay, so what I hope that I've convinced you of, or at least that you have an understanding of now, is that, number one, ALS is a syndrome. Arthur Hudson told us that almost 25, 30 years ago now, right? He said that about 15% will have a dementia. What we're saying now, we know, is somewhere between 50 and 60% will have some aspect of a frontotemporal spectrum disorder. When it does happen, though, it's not a curiosity. It is prognostically important. It helps us in understanding the disease progression, and particularly if there's behavioral or executive dysfunction. This is critical. And this 175 tau story has turned out to be critically important because it is the modification that is causing this. We now know it from cell culture as well as in animal models, and we know that we have a panel of drugs that should allow us to discontinue this. So, at the end of the day, all of this work is done by a tremendous lab group. This is that group. I think if there are any questions or comments, available to do so. And this, I hope everybody knows what this is.
Thank you so much, Mike. Thank you for that. We really appreciate it. That's a wonderful overview, exactly what we were looking for. We do have a number of questions. Can you hear me? Just making sure.
I can hear you well, yes.
Okay, perfect, perfect. Okay, so so I'm going to start with a question actually that leads in, kind of merges one of the questions we got from a viewer with one of my own, and that was I was at a talk in Dublin where they had 3 neurologists who were doing a panel, well-known ALS neurologists, you'd know them all, and they asked a question about what would the top 3 things, you could only choose 3 things to test someone for when they come into your ALS clinic, what would they be? And all 3 of them, one of the top 3 was looking at a cognitive screen of some sort, some form of screen or test. So I was wondering, like, there's a lot of research being done about different tests, the ECAS, people using mini-mentals, and various other cognitive screens. What would be a good example of one that someone could use in their clinic easily and could be a reasonably verifiable test?
Yeah, that's a great question. You're right. I bet you I can absolutely guess who the 3 individuals are and why they came up with that answer right away. So first off, one of the things that we're looking to do is to make sure that there is an easily used single bedside test. We don't have it yet, although we have a number of indices of tests that would be helpful for us. So I'll tell you what I do, and it works to kind of open the door. So I use something called the MoCA. It's the Montreal Cognitive Assessment. It can give you a fairly robust score in studies that have been using it, looking in ALS. It has a number of features that are well represented and do pick up the early changes. If I was in Europe, I would certainly use ECAS. I think the study out of Edinburgh and that group with Sharon Abrahams and her colleagues has been excellent. So there are a couple of easy bedside studies to do. Doug Munoz, one of our colleagues who you'll know well, and many of you on the screen may know, out of Queen's University, has done some beautiful work, really careers work, looking at eye tracking and something we call saccadic versus anti-saccadic pursuit mechanisms. And what we know is that we can easily test eye movements at the bedside by simply having an individual look from a fixed point over to a second point without turning their head but only moving their eyes. And as a neurologist, we can watch that and we can see, is there hesitation to it? Do they go to the wrong direction? Is there a slowness in which the eye moves across and such? Is there a degree of persistence, what we call ocular persistence with that? So a very simple 5-second test, and I would add that to the MoCA as well. And then the final thing is that we have a number of bedside things that we do on our physical examination that are called frontal release signs. So physical signs that, such as just stroking the palm of the hand and looking for a movement around the chin. Things that you'd wonder, geez, why does a neurologist do that? Well, when we do it, we're looking to put them together as part of a package. So for me, I do those 3 things, and if those are abnormal, then immediately I'm starting to have some questions as to, okay, I think I need to go a step beyond all of this. And that's when I bring the imaging in, and that's when I bring more formal studies in.
Fantastic. Thank you for that. So another thing that you brought up earlier was about how, a very recent review on ALS didn't really even mention anything to do with the spectrum or to cognitive dysfunction. And I just was wondering, like, even now we have pure genetic evidence that there are families where they have frontotemporal syndromes, they have ALS, and then they have both in some individuals. I just wondering if you could sort of mention about the resistance in the field that has been over the years to accept this and how it's gotten better but maybe still isn't there.
Yeah, so I'm going to use a quote from a well-known politician who happens to head the American government and say simply, so sad. At the end of the day, it's an important piece that you're highlighting there. When I remember when we were first starting to do an awful lot of this work, and there were investigators in Germany who had done some of the very early studies using what's called HMPAO spec studies, so a way of actually using a metabolic measure of the brain. And he actually came and lectured at one of our sessions and showed the original slide that showed that there were regions of the brain that had a reduction in perfusion in ALS patients, and it was outside of the region of the motor system. This is Albert Ludolph's work. And Albert, that paper he sent to Brain, which is one of the preeminent journals, and they rejected it immediately. And basically said that there's no evidence that in ALS that there would be any aspects of dementia, and therefore the study must be flawed for it. Now here we are, decades later, and Albert was bang on with all of that. When we submitted our first papers demonstrating that there was a tau deposition in ALS, so this would be 2001, in that time period, the reviewer came back and said, well, we believe the evidence is here, but I'm not sure that we could live with ourselves if there's evidence that ALS patients have a dementia for it. So the paper was rejected on that basis alone for that. And yet families, when we were starting to have the conversation that this existed, were uniformly in the opposite direction of going, now that explains something. So the scientific community comes from an unfortunate position that we often find ourselves in of being well beyond or well behind where science is taking us and refusing to accept what's actually there. You're right, the genetic data is there's no way of arguing with it. It's solid. The pathology is solid. And so how we can still have conversations and not include the two together, still, it is so sad. We're getting there, though. It's getting better.
So, thank you. So maybe one more question, and it comes down to, so you were talking about how the survival curves can be different for individuals who show different forms of cognitive impairment. It's heartening to know, at least based on what you just said, that the most recent clinical trials guidelines that were developed for ALS have included that you should be screening for cognitive impairment in terms of at least stratifying your data to make sure that you're examining that. But in terms of therapeutic overlap and thinking about, lithium, for example, do you suspect there's a potential that we may have therapy that will work for individuals who have both the classical ALS and more of a spectrum type disorder? Or do you think we might have to start thinking about pairing things where we're treating, one versus the other and maybe seeing if they work together or complementary, especially since there's different neurons at play here, not just motor neurons, which a lot of us are studying in the field, but also other neurons, as you mentioned, hippocampal and others.
Yeah, so it's a great question, and it's probably one of the most fundamental ones that you can ask in there. So I think all of us at the end of the day would love to see that we can unite these pathologies into a single mechanism. I think we're a little ways away from that yet. When we're there, then I think we might be able to have the conversation around a single drug or a single intervention, which will have an impact on both aspects of it. But I can tell you, as a clinical neurologist, to me it doesn't matter. What really matters is that we have multiple disorders that we treat using a panel of medications and many medications simultaneously. So for instance, an individual with Parkinson's, as the Parkinson's evolves over time, we will often be using 2, 3, 4 medications simultaneously for that, some affecting different processes, some augmenting the effect of one drug over the other for it. So I think we need to be prepared in the ALS field that we are actually going to be looking at more than one drug therapy simultaneously. And at the moment, I think the travesty would be is that we find a treatment for individuals on the motor side of things and the cognitive progresses, or vice versa for it. So I think what we really need to do is to kind of accept right now that we're going to need to have 2 drug therapies at minimum to really deal with this disorder. One on the cognitive side and the second on the motor neuron side.
Great. Well, thank you so much. Thank you for your time. Thank you for the wonderful talk, and We really, really appreciate it, Dr. Strong, and we'll talk to you again soon.
Great. Thank you very much. I've really enjoyed this. Take care.
Thanks. Take care.